This is podcast 10. As I mentioned in podcast 9, microtubules, microfilaments, and intermediate filaments form part of the cytoskeleton. In fact, form all the cytoskeleton. We'll go into those in more detail in podcast 11, 12, and 13. I just want you to keep in mind for this podcast when we're talking about how these membrane vesicles move throughout the cytoplasm, they are moving within this dense cytoskeletal network, this protein meshwork, so they're moving within the soggy nerve ball. Okay, here's a question for you. Okay, so we're going to talk about the endomembrane system from the ER to the cis Golgi, through the medial Golgi, trans Golgi, and then eventually out to the plasma membrane, maybe stopping off at a lysosome. And there's this little animation I want to show you.
The membrane system consists of the endoplasmic reticulum and Golgi apparatuses.
Endoplasmic reticulum, Golgi.
Vesicles pass from one part of the endomembrane system to another, and secretory vesicles from the Golgi apparatus merge with the plasma membrane, secreting their contents by exocytosis.
So you have the cis Golgi network, which is CGN facing the endoplasmic reticulum. Medial cisternae are in the middle. Trans Golgi facing the plasma membrane. And the default pathway is from the rough endoplasmic reticulum to the plasma membrane. How they have figured out a lot of this stuff is by labeling proteins that are made on the rough endoplasmic reticulum with a radioactive amino acid, and then taking images with cells at different stages at different time points to see where are those cells. So if you label a protein that is being made on the rough endoplasmic reticulum and then wait for 3 minutes and take a picture, you get most of the protein, which is this squiggly stuff here, is found inside the rough ER as it's being synthesized. That's after 3 minutes. Then after 7 minutes, most of the protein is now moving to the cis Golgi. Okay, showed in here, kind of hard to tell on these diagrams, but you can take my word for it here. And then after 37 minutes, it's moved out of the Golgi into these zymogen granules, which are the condensing vacuoles next to the Golgi on their way outside the cell. And then 117 minutes, they're in the zymogen granules, in the vesicles that are going to be fusing with the plasma membrane and headed outside the cells. So these are protein-packed vesicles between the trans Golgi and the plasma membrane. These are vesicles that are going to fuse with the plasma membrane and deliver their contents outside the cell. So that's how they figured out this pathway. I just want to mention that there is a physiological phenomenon that's associated with a lot of different diseases called ER stress, in that the cell is basically taxing this pathway too much. Too much of this conveyor belt from the rough ER, cis, medial, trans, Golgi outside the plasma membrane. Too much of this is happening at too great a rate, and it puts a stress on the cell, and in general it's called ER stress. So if you run into that term in a paper, that's what they're talking about. So at the cell membrane, what happens is you have the secretory vesicle. In the electron micrographs, they call these zymogen granules. They are packed full of proteins, and they have membrane-bound proteins in the membrane itself. They're going to fuse with the plasma membrane. That plasma membrane is now going to become part of the vesicle. The vesicle is going to become part of the plasma membrane. So the plasma membrane gets a little bit bigger by adding the vesicle to it. and then the contents of the vesicle leave the cell into the extracellular fluid. If we're talking about a vertebrate, this would be blood or lymph, I guess. The other way can happen too, where the cell will form a pocket and incorporate things from the outside environment. This can be non-specific, which is just pinocytosis or just general endocytosis, or it can be specific, where something binds to a receptor first and then forms a vesicle. We'll talk about that later. That's called receptor-mediated endocytosis. But nonetheless, a vesicle forms by pinching off part of the, or invaginating part of the membrane, and then eventually pinching it off. There's a protein here that is involved in cutting off that last little bit of the membrane. It's called dynamin. And then the vesicle becomes free of the membrane and free to travel to the interior of the cell. So it can go in either direction, exocytosis or endocytosis. Okay, H5P question. So once again, we have this default pathway from the rough ER to these transition vesicles going to the cis Golgi network, and then through the medial Golgi, if you're going in the direction, this direction, toward from the rough ER to the plasma membrane, that's called anterograde transport. And then after it gets out of the trans Golgi, forms these secretory vesicles in those micrographs. We call them zymogen granules, and then fuse with the membrane, exocytosis releasing those components. Endocytosis can also occur, where you just form a pocket of the membrane and capture what is ever outside the cell. Then the vesicle will be fusing with a low pH compartment called an early endosome. That early endosome is going to become a late endosome. So what can happen is these can already exist in the cell, or proteins can be added to these vesicles so that they become this. And the same thing happens to a lysosome. So you can fuse with an already existing lysosomes, or you can import proteins into this compartment to turn it into a lysosome. Add lysosomal proteins to it, and then it becomes a lysosome. These compartments are, okay, so in the endocytotic pathway, first you form these vesicles and then they detach from the membrane. They, from an early endosome can become a late endosome. This is just how far along you are in the maturation of the vesicle, how low is the pH in this vesicle. And then it can fuse with the trans Golgi network and return to the ER. There are systems that have proteins that need to stay resident in the ER in order to work. And every once in a while, those proteins will escape, because this is the default pathway, and they get caught in a vesicle and they end up on the cell surface. If that happens, there are systems at the cell surface that can return them back to where they're supposed to be, and this is that retrograde transport here, so it returns it back to where it's supposed to be in the rough ER. And or if what you're bringing in needs to be disassembled, then you can do that in a lysosome. That squeaking in the background is my puppy playing with her toy. Okay, so a question is do these membrane vesicles stay put as a place inside the cell, and the vesicles move into or out of that thing, or does everything move as it matures or develops? So is it that the vesicles move and fuse into this thing that stays put, or does cisternae become medial, which then becomes trans? Is this physically moving this way, or is it that that these stay put and then the vesicles fuse in and out? There's evidence for both types of systems in different kinds of cells. So there's evidence that the membranes stay put and these vesicles move things from one set of membranes to another. And there's evidence in other kinds of cells that the membranes actually move, and that this becomes that. Okay. I suggest when you're studying that you practice drawing a simple diagram that has the endomembrane system. What's the default direction? What's the default fate of a protein that is translated on the rough ER? And you explain it to someone else. Maybe somebody in your group, or your dog, or your grandma, anybody else. Anything that you can do that is active is going to help you remember it more than just listening to a podcast or just watching a video. In the default pathway of going from the ER to the cis-Golgi network, medial cisternae trans-Golgi network, there are proteins and functions that are specific to different compartments, and there are mechanisms for keeping proteins in those specific compartments, even though the default pathway is that you keep moving all the way to the cell membrane. So, for example, in the ER, we know that the carbohydrate that is going to be added to the protein to modify it in the Golgi starts to be synthesized in the ER. And anything that is not properly folded will be removed. And we know that, for example, by the trans-Golgi apparatus, if there's going to be a sulfate added to tyrosine, that is added in the trans-Golgi apparatus. So there are things that are specific to these different compartments and proteins that perform these functions. And like I said, there are mechanisms for keeping those proteins out of the stream that is moving this way so that they stay put. Okay. I mentioned this in another podcast, how you have a problem with lysosomes that are full of digestive enzymes, proteins that digest proteins, and the whole system is largely protein. So how is it that you can make a protein that digests proteins without destroying the system? And I use the example of mannose-6-phosphate. This is just more detail on that. So as this protein that's going to be a digestive enzyme of other proteins is made, it has the carbohydrate mannose attached to it, and then immediately it's phosphorylated. That phosphorylation prevents it from being active, and that phosphorylation is recognized by a mannose-6-phosphate receptor in the trans-Golgi. That mannose-6-phosphate receptor picks up those proteins that are then fused with a late endosome, which then will become a lysosome, which will remove that phosphate and then allow that digestive enzyme to be active. So with the phosphate on it, it's not digesting proteins, and the low pH environment of the lysosome allows the phosphate to come off and for it to become active. And it doesn't become active until it's in its compartment where it needs to work. In terms of in the ER synthesizing those carbohydrates that are going to get added onto the proteins in the Golgi apparatus, this is just an example of the detail of how those carbohydrates would be made. So a dolicol molecule that's within the membrane of the ER gets modified with a carbohydrate, which gets another subunit added onto it, and then several more subunits added onto it. Then there's a flipase that moves the building carbohydrate from the cytosolic phase to the ER lumen face, it gets modified several more times and eventually is going to attach itself to a protein in the Golgi. So do you have to know the names of all these things? No. You will eventually ask the question, so like how much do I need to know? And the answer to that question is anything that we talked about in these podcasts is fair game. And I will ask you questions at the same level that I talked about it in these podcasts. So did I ask you, did we talk about the names of all these intermediates? No, we did not. Did we talk about the specific structure of all these intermediates? No, we did not. So I would not ask you that. But I might ask you, what is the name of one of the starting molecules to build a carbohydrate addition onto a protein, and you would say dalacol, for example. Okay, as I've said before, we're trying to give you some context for how this relates to the fight against the coronavirus. And in one of the first podcasts, I mentioned that the spike protein, that is what the coronavirus is used to attach to an ACE receptor and to gain entry into the cell, that spike protein is very heavily glycosylated. All these fuzzy purple and green things on this model are all carbohydrate additions onto that spike protein. And you can see it changes the shape of that protein pretty dramatically. And so if we are making antibodies against this thing, and a lot of the virus vaccines are targeted against this thing, you have to consider that this thing is very heavily glycosylated and not just consider the protein part, but also the carbohydrate part, especially if an antibody is going to recognize it. And this carbohydrate addition happens in the Golgi as the virus is using the cell's own machinery to make more copies of itself. So it's making copies of the spike protein. Those copies are being processed through the Golgi where these carbohydrates are being added. So one possible intervention could be a drug that prevents carbohydrate addition. Of course, you'd have to say, how would that be specific to the virus and not proteins we do need? And that would be a problem. In fact, there are drugs that prevent carbohydrate addition in cell culture, but they tend to be super toxic because your cells need those carbohydrate additions. Okay, another H5P question. When cells are endocytosing, it can be more specific. It can be just general, taking in fluid. It's called pinocytosis. There can be a more specific receptor-mediated endocytosis. In this case, this would be something like a macrophage endocytosing a bacterium. There would be a cell receptor that would recognize proteins or carbohydrates on the outside of the bacterium. They would bind. That would cause the cell to extend what these false feet or pseudopods around that bacterium, eventually fusing on the other end of that bacterium and then engulfing that bacterium in a vesicle. And then that vesicle then interacts with early endosomes and then eventually a lysosome to break apart and destroy that bacterium in, say, like a macrophage. And receptor-mediated endocytosis is a big theme in cells, and there are many drug interventions that are based on this system. So, for example, the cholesterol-lowering drugs take advantage of this system. They're actually involved at a different part than is on this diagram, but they involve this sort of system. So the idea with receptor-mediated endocytosis is there's a specific receptor, which is a protein in the cell membrane, that it binds a specific ligand that would be floating around in the blood. For example, it could be a low-density lipoprotein particle and the ApoB band around that particle. the thing that carries cholesterol in your blood. Once those particles are bound to their ligands, they are gathered through fluid mosaic membrane. They move laterally through the membrane, and there are proteins called assembly proteins, assembly particles, that will start to form what's called a coated pit. This is a depression in the membrane that's eventually going to become a vesicle. On the inside side of the membrane, there are units of protein that are going to coat this vesicle, and by polymerizing around this pit, they help to form the vesicle. So they actually pull in on the membrane, sort of like if you had your mouth on a balloon and you were sucking in. These polymerizing of these proteins help to suck in that membrane, and eventually you form a vesicle. Here's that protein dynamin that we talked about before. It's a donut-shaped protein that's going to cut off the neck of that last little piece of membrane and form a vesicle that is now coated with a protein. In this example, it is coated with clathrin, and the protein will then uncoat, which is a signal for the vesicle to fuse with an early endosome. So after it's coated, it becomes uncoated. The clathrin gets recycled, can coat other vesicles. It fuses with an early endosome, which will eventually fuse with a lysosome, and whatever was taken in will be taken apart into its macromolecules. At the same time, the receptors are recycled. They form another vesicle, fuse back with the membrane, and you can do the whole thing again. So this is called receptor-mediated endocytosis. There's a coronavirus connection here too. So this drug, chloroquine, hydroxychloroquine, became famous in sort of a political football because President Trump wanted to promote this drug. However, it did not have good results in clinical trials and is now not recommended. So the mechanism for chloroquine, hydroxychloroquine to work is that it increases the pH of these intracellular vesicles. So in an intracellular vesicle, when it fuses with an early endosome, the pH inside these vesicles is around 5, okay, in these vesicles, in these endosomes and in the lysosome, versus the pH of the cell is about 7. So that's a big change in hydrogen ion concentration, and usually that change in pH, when the receptor gets fused into that low pH environment, that causes the receptor to change shape and for it to let go of its ligand. So the idea with using that drug was that when the coronavirus was endocytosed into the cell after binding to the ACE receptor, that it would fuse with an endosome. There'd be a pH change, and that would release the coronavirus into the cell, and then the ACE receptors would go back up to the cell surface. Hydroxychloroquine or chloroquine can increase the pH of these intracellular vesicles, preventing that release of the coronavirus from its ACE2 receptor. However, it didn't work, and it also caused some serious heart issues for people that had pre-existing conditions. And in the clinical trials that were done, either there was no difference or people ended up with a worse outcome, more deaths if they were on the treatment than if they were not on the treatment. So it's not used anymore, or not recommended anymore. These are just some electron micrographs to show you what receptor-mediated endocytosis looks like. So here is the bound ligands, here's the coated pit, and here's the coating on the inside of the pit. Further along in the development of the vesicle, here's where Dynamin comes in to cut off that vesicle, and then the free vesicle, still with its coating, its clathrin coating, after it's been endocytosed. Clathrin is not the only protein that can coat vesicles. There's lots of different coating proteins. They have different names. COP just stands for coating protein 1, coating protein 2, etc. Some of these are specific to which direction you're going and what destination you ultimately have, but there are many different ones other than clathrin. Clathrin is a particularly cool protein and famous because it has this triskelion subunit. So this is each individual subunit of clathrin protein looks like sort of a bicycle wheel spoke, and then they polymerize together into this geodesic dome sort of shape. And they share a name with the Marvel Cinematic Universe base for Agents of S.H.I.E.L.D. Yes, that is how much of a nerd I am, that I thought you might know that. Or that I know that. Anyway. So when you put a whole bunch of triskelion units together, you get this sort of structure. This is an electron micrograph. This is an electron micrograph that actually looks like a little geodesic dome inside your cell. And this is the protein coating the outside of the vesicle. So as they polymerize, they help to form that sphere, that vesicle, and then they can depolymerize and go off and form another coated vesicle. You have a whole bunch of vesicles fusing together. How does the cis vesicles know to go to the medial? How does medial know to go to trans? How does the rough ER know to go to the cis Golgi? A lot of that is governed by snares and snaps. So snares and snaps are yet more proteins. They're on the outside membrane of these. They're membrane bound and an outward facing membrane of these vesicles. And they have to agree with each other for a vesicle to properly dock. So here you have a V snare, and it's compatible with another receiving protein that is another part of this snare snap system. So these proteins have to come together and agree for the vesicle to fuse and release its content to wherever it's going, say it's a lysosome or the rough ER. And if they don't agree here and there, then they are then the vesicle will bounce off and it will find its correct component. Botox, botulinum toxin, is a protease of snare, these proteins here, and so it digests these proteins and prevents this from happening. So it can prevent muscles from contracting as a result, or I'm sorry, keep muscles in tetanus. So people who have Botox injections have that permanent look of surprise on their face. Their muscles are very, very tight because their snares have been proteolytically cleaved and prevents nerve transmission, keeps those muscles in tetanus. Okay, lysosomes are going to be the ultimate destination of large packages of things that come from receptor-mediated endocytosis and enter the cell. They're going to be taken apart in lysosomes. Also, old organelles also fuse with lysosomes and get degraded. Here's just a picture of a mitochondrion on its way to the mitochondrion graveyard, and then these are vacuoles or remnants of mitochondria after they have fused with lysosomes. If it's a protein that gets worn out inside the cell, that's degraded in a proteasome, not a lysosome. So these are just the different places from which lysosomes can receive input. And so what's the difference between an early endosome, a late endosome, and a lysosome? It's just maturation of the organelle. So the proteins in an early endosome and a late endosome and a lysosome are not going to be exactly the same, but you're just going to turn an early endosome into a late endosome by adding those proteins to an early endosome. And you're just going to turn a late endosome into a lysosome by adding those proteins into the late endosome. And then it becomes a lysosome. Okay, one more last H5P.